In this video, we will continue our discussion about this hGTSE-1 and p53 interaction, and this time we will discuss about this interaction towards preventing apoptosis in the S and G2 phase of the cell cycle. So in the previous videos, we have discussed about their interaction to prevent apoptosis in general within the cells. However, in this video we will discuss the interaction within the S and G2 phase of cycle, or prominent in S and G2 phase of cycle. So first what they did is they separate the cells through flow cytometer into different cell cycles, that is in cells that are rich in G1 phase and cells that are rich in S and G2 phase, and then they knock down hGTSE and see its impact on p53, so that they can be positive about their hypothesis before moving further. So in the G1 phase, when they check about the effect of hGTSE knockdown, and they can see that there is not much effect they have observed, or almost no effect they have observed in the p53 production. Even though the hGTSE is knocked down, they should see tremendous increase in the p53. However, there is slight increase in the p53 that is observed. When they check this cyclin A, which is usually considered as the antibody for S and G2 phase, it's also very low. So this indicates that the separation of G1 and S and G2 phase is uh, happened to a greater extent, or enrichment of cells in G1 and S and G2 phase separately. Separation of G1 phase cells and cells that are in S and G2 phase happens uh, efficiently, if not 100%. Or so let us now continue our discussion. So for this S and G2 phase, when they knock down the hGTSE cell, they can see the tremendous increase in the p53. So from this, they get a positive confidence, or that this hGTSE-1 and p53 interaction happens mainly in the S and G2 phase of the cycle. Later on, they develop a strategy to compute the and apoptosis cells in the S and G2 phase cycle through these two dye. First one is the cyclin A dye, another one is the propenium dye. So what they did is they stain the S and G2 phase cells using these two dyes, that is cyclin A dye and the propenium dye. And this green color shows the cyclin A symbols, and for this cyclin A image, this is the counter propenium dyed image, and this arrow indicates the apoptotic condition of the nuclei. And we can see that in all so this is how they used to compute this. First they test whether the cells are in S and G2 phase or not, and if they are in S and G2 phase, then they check whether their nuclei has a apoptotic condition or not. So in this case, the cells are not in S and G2 phase, since they are not positive for cyclin A. However, these cells are apoptotic condition. Whereas in this condition, in suppose this seventh number figure, cells are in S and G2 phase, since it is cyclin A positive, and same cells are in apoptotic condition, as it is shown by propenium diodide, nuclei of the propenium iodide, nuclei of the cells with the help of propenium iodide. So this is the computational strategy to check whether cells are in S or G2 phase, and whether these cells are apoptotic condition, or these cells are in apoptotic condition or not, through proponium iodide. So they did their analysis on the control cells, or we can say that on the control cells and cells that are cells in which hGTSE is knocked down. And so after that, what they did is they check, or they separate take down the number of apoptotic cells in both the condition. So these are the total number of apoptotic cells that are mentioned here, and this black dark you can say the black color indicates cells that are positive for cyclin A, and here also the cells that are positive for cyclin A, and we can see that the cyclin A represents, this is, you know, that cyclin A represents the cells that are in S and G2 phase. So in control condition where hGTSE is knocked down, number of apoptotic cells that are in S and G2 phase are low, whereas in this S condition, second condition in which hGTSE-1 is knocked down, the number of positive cyclin A cells are more. That means that this number of apoptotic cells are mainly from S and G2 phase, and this is in align with our understanding that hGTSE and p53 works mainly in S and G2 phase cycles. That's why the knockdown of hGTSE will result in more apoptotic cells in the S or S and G2 phase. Later on they also check whether these signals are not because of the rise in the amount of cyclin A within the cells because of some interaction of hGTSE. So in this they check in no stress condition and also in stress condition, and they can see the same amount of cyclin A within the within all samples or all conditions. So this indicates that the hGTSE does not interfere with cyclin A production within the cells. And all of this figure and combining all of these experiments, they later on conclude that this hGTSE and p53 interaction control apoptosis mainly in the S and G2 phase of the cell cycle. So if you need to understand this story or these figures in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.